So welcome to IMRI's first ever bite-sized webinar. Thank you so much for joining today. We're really happy to have you here. So my name is Kate. I'm the Community Engagement and Events Coordinator for IMRI. At IMRI, due to COVID, like a lot of organisations, we've had to take our events online. So please bear with us. As I said, this is our first one, so hopefully we won't have too many technical problems. We've called these sessions a bite-sized science webinar because this is just designed to give you a little taster of the type of research that we have going on here at IMRI. And for those of you who don't know, IMRI is a not-for-profit charity. We support health and medical researchers across the Illawarra Shoalhaven region of New South Wales. We bring scientists, doctors and health professionals together to solve our greatest health challenges at the moment. We do research into all different types of diseases like cancer, diabetes, mental health and dementia, just to name a few. Our region's population is incredibly diverse. We have some established areas with an Asian population, but we also have newer areas with a much younger demographic. There are affluent areas, and there are also areas of socioeconomic disadvantage. And as a result of that, the Illawarra Shoalhaven region provides a perfect snapshot of the health and well-being of Australians. So really, this is a perfect area to do health and medical research. And before we get stuck into it, I also want to say a huge thank you to all the staff, students and researchers at IMRI who have worked really hard during this pandemic to adapt and make all the necessary changes that they need to do in order for the research to continue and also the teaching to continue. We know it's been a real there's been all sorts of different challenges, so we just really want to acknowledge all your hard work and thank you for sticking with us and supporting us. And of course, I'd like to thank our supporters, IMRI supporters, our community groups, our support groups, and our donors, all those people who ensure that IMRI can keep doing what we're doing. We're really grateful for your support. So I think what we'll do now is get started, and I'd like to hand you over to Dr. Luke McAlary. Luke is a passionate researcher. He enjoys the challenge that science presents. Luke's research focus is on the role of protein aggregation in neurodegenerative diseases. Specifically, he's interested in ALS, MND, and is trying to determine why protein aggregation causes this disease and how it could be stopped. He is a postdoctoral fellow currently working on investigating a novel gene implicated in ALS MND pathology. Was that right, Luke? Did I introduce you correctly?
Yeah, thanks for the kind introduction, Kate.
You're welcome.
So am I taking over?
Yes, please.
Awesome. All right, hi everyone. Thanks for taking your time out of your day to come watch this sort of thing. I'd recommend watching some of the other webinars that IMRI's hosting because they're quite good too. And I know a few of the researchers doing that, and they'll be pretty good to watch. So I'll just take a moment to share my screen here. So. All right, so as Kate kindly said, I work on Neurone Disease, or Neurone Disease or ALS, and I work at this from a background of basic science, moving into translational science, but I can explain that later. So a question even after the ICE challenge, which you may have remembered from a couple of years ago, people still have, is what is motor neurone disease exactly? So I'm going to give you a really basic background to that. And so let's just jump right into it. So first of all, affected by motor neurone. Well, it's a wide range of people, but it's mostly people in that 50 to 60 year range who are affected by motor neurone disease. There's only around about 1,200 people in Australia living with MND currently. There's 400 new people diagnosed this year. And you might be wondering, how does that stack up? Well, the reason that there are so few people living with MND is because it is a terminal illness that progresses quite rapidly. So even though we get 400 new cases per year, a lot of the people who have it unfortunately pass away. And this is why there is an urgent need for more research and us to find more effective treatments and cures for this terrible disorder. The majority of people who are diagnosed with MND, there is no familial history of disease. It is what we term sporadic. But around about 1 in 10 cases are familial, meaning that there is a family history and that there may be a known genetic cause. And so I just want to show you this plot here. And so whilst it may not mean a lot to you, it means a lot to us as researchers. And what you see here is the calendar year and then a number of genes. And this is how quickly we as researchers had moved since the first gene found to be associated with ALS to identifying many, many, many more genes. And whilst this tells us that it's quite a complex disease, there's also something very cool to be seen about this plot in that we are moving very quickly to understanding it to its core. And this should be a great cause of celebration for people because we are moving very rapidly. So what is the disease? Well, if we look at the human in a very simplistic fashion here, they have a brain and a central nervous system that innervates their musculature so they can move around. So it's actually a neuromuscular disease or neurodegenerative disease in which these motor neurons that innervate your muscles degenerate and die. And so what happens is you lose the ability to move and eventually the ability to breathe. And this is how most people pass away, unfortunately, from this disease. And major point, currently we have no cure or effective treatments for this disease. And so a lot of research is being put forward towards this and trying to get better treatments and maybe potentially even one day finding a cure. And the main feature of this disease is the generation of dense protein clumps inside the cells. And so if you think back to high school biology of what a protein does, well, most of your body is composed of proteins. And these are the tiny little molecules that take action to achieve outcomes for your cells. So we come at this from a question of why do proteins clump in the first place? And again, if you think back to high school biology, you might understand or remember that your proteins need to fold into a 3D shape. So when they're initially generated inside the cell, they're a loose noodle almost. And what you have here are these little orange spots in the protein. And these are hydrophobic. They don't like being around water, so they want to be around themselves. And so normally what would happen is the protein would fold so that those hydrophobic components, those sticky parts, are shielded from the surrounding water inside your cell. But what can go wrong is your protein can become misfolded. Doesn't reach this nice shape. It unfortunately becomes a little bit misshapen. And then these can start to clump together into what we call aggregates or aggregated proteins. And this is happening all the time in me, you, kids, old people, everyone. And unfortunately what happens is as someone ages, their body and their cells' ability to deal with the aggregated protein declines. And so if you have a mutation, this can be exacerbated even further. So how do we look at this in the lab? Well, we take a very basic but very cool approach, I think, to doing this. Really what we do at its core is we exploit the code of life, DNA. So the central dogma of biology is the ability of DNA to be turned into protein by your cells or by any what we consider a living organism. And so one of the ways we can do this is we can exploit even bacteria to do this for us. So bacteria are a very simple organism and we know how to manipulate them very effectively. And so by inputting DNA that codes for a certain protein, we can force that bacteria to generate our protein and become tiny factories for us. And then after the bacteria has done what it's needed to do, we can extract the protein. And the reason we want to get that protein extracted by itself is so we can look at it by itself and turn some very powerful tools onto it. So something we can do with a protein we've extracted from bacteria, say an MND protein, is we can look at the level of clumping that occurs to the protein or aggregation across time. And we can see here that a mutant protein will clump much, much quicker than a normal protein. And an additive to that, we can start to ask questions about, well, we think maybe this drug, this chemical might prevent that. And we can actually treat the protein with a chemical to prevent its clumping. We can also leverage some of the much more powerful technologies we have, such as an electron microscope, to actually look at the clumps that are formed. And even though we call them clumps and we think of them as these disordered, weird looking things, in fact, there can be some order to them. And so what you're looking at here are these long fibers composed of an MND protein called TDP43. And so this is actually like polymerization rather than random clumping. And this tells us something about the disease. And so what I've drawn here is the misfolded protein and an arrow pointing to its actual size within these clumps. So you can see it takes a lot of misfolded protein to start to form these worms that exist on this in this sample. And so one of the other things we can do is we can look at this in cells. So this is outside of bacteria, but in human cells. And one of the things we need to do in this case is we need to be able to better see what's going on. And so it's hard to see individual proteins in a cell because we don't want to remove them like we did from the bacteria. We want them to be in the cell doing what they normally do. And there's a lot of proteins in your cell. So one of the ways we do that is, again, we exploit the code of life, DNA. And so jellyfish can glow in the dark, and we can take that DNA, that protein that lets jellyfish do that, and tag our proteins with it, and they'll light up. And so that's what this might look like in a very simplistic manner. We now have our folded protein with the attached glowing fluorescent protein here. And so again, done by exploiting DNA. And so this image is basically what you'd see if you used a microscope to look at a bunch of cells growing flat on some glass. And there's not really much you can tell from that, right? And I don't expect anyone to tell anything from that. You can see some cell borders and maybe some nucleus of the cells, but there's not much going on. But if we have the glowing protein in there, we can start to make out a lot more information about what's going on. And so what we're seeing here is the cells with the glowing protein in them, and also these dense areas within the cells that are the protein clumps, right? And so what we're seeing here is that even in this very simple cell model, we can recreate the disease from humans. And this makes this now similar to the protein from bacteria, but an even better way of looking at this system and trying to treat it. And so one of the ways we can do this is using maybe that drug we were talking about before. And so what you're seeing here is a video of the cells growing over time with several drugs put into them. And you can see where we have no drug, we have a lot of clumping going on in these bright spots. Drug 1, not as effective, but then drug 2 and drug 3 showing some promise. And this is one of the ways we can start to formulate and find out new effective, maybe more effective treatments for MND in the case of certain proteins. And this is something we currently do in our lab. And so since this is more of an informal affair, I want to end the presentation a little bit more quickly and tell you a little bit about what we're currently trying to do in a very summarized format. So we're trying to make better systems to examine protein clumping. We're still investigating the role of protein clumping as a cause of MND because it's quite a very heterogeneous disease, very confusing. And we're also testing quite quite large scale new drugs and therapies that block protein clumping. And so where we sort of fit into this is we do a lot of what's called basic science. This is very simplistic science, very hypothesis-driven science, trying to understand the disease. And then we move into this applied science realm where we try to apply maybe what we understand about the disease to form more effective outcomes for people with disease. And with that, I'd like to thank all the people that have put forward into not only the projects and the ideas I showed there, but the people who've helped me throughout my career. So you have Justin Yerbury in particular, Professor Stephen Plotkin from UBC, and also Professor Neil Cashman from UBC, who have been my sort of mentors throughout my early career. And with that, I'm open for questions now.
All right, thank you so much, Luke. That was a fantastic explanation of what you're doing in the lab there. So does anybody have any questions they want to send through? You can pop those in the Q&A box and I can pick those up. We've got one here that's come through. Luke, this has been a really challenging time for a lot of people, and researchers have not been excluded from that. Can you just give us a little bit of information on how has COVID-19 impacted your research, if at all?
Mainly, it's impacted our research in the form of increased social distancing in the lab has been a bit of an issue, because oftentimes you're working in close proximity to other people. So we have to be aware of that. We have to work away from people. And a lot of the time, we're not sitting in an office, we're in the lab, hands-on, trying to work at a problem that might take, 10, 12 hours of you just sitting there. You need to work at it, and you've got to continually be aware of those working around you. It's been very difficult getting our hands on some of the reagents that we need. So for growing cells, drug treatments that we want to test, difficult to source because of supply line issues. And also our lab operates, or I operate in Justin Yerbury's lab, and Justin has unfortunately been very difficult for him to come to work and see us and stuff like that. So it's been difficult for us to be a little bit more distanced from Justin when we can, sometimes be in a meeting room with him. That's now not easy to do. So it's similar to what most people are dealing with, really, just having to be aware of what's going on around you.
That's it. So just make sure that everyone that's in the lab is safe.
Yes. Yes.
So we have a question here from an anonymous attendee. Does your research affect or involve other diseases? And I think what the question is trying to get at is whether the outcomes that you've had from your research, has that had an impact on treatment or diagnosis pathways for other diseases?
Absolutely, that's a really good question too. So a lot of what we do, we focus on MND, but MND is considered to be a disease that exists on the spectrum with another disorder called frontotemporal dementia. So one of the main proteins we look at in MND, which is called TDP43, is also involved in frontotemporal dementia. So it's expected that some sort of treatment or effective compound that may work against TDP in MND may also have a crossover into FTD, frontotemporal dementia. But likewise, there's a lot of disorders that involve protein aggregation or clumping in general, such as Parkinson's disease, Alzheimer's disease, multiple system atrophy. All these involve a similar concept where a protein will clump. And perhaps some of the findings we have in MND will cause ideas to happen in those fields. Because I know I've definitely been influenced by Parkinson's research and Alzheimer's research. So of course, yes, I think that's a thing that will happen.
Great, thanks for sharing that. Another question that we have here. So what does your day as a researcher look like? How much time do you spend in the lab? Do you spend just as much time at your computer? You've already mentioned that you've been influenced by Parkinson's disease research. How much do you chat with other researchers within IMRI and perhaps elsewhere as well?
So I typically get to work early in the morning, around 7 o'clock, maybe put an experiment on, grab a coffee with the coffee people, and go back into the lab. And it sort of fluctuates how much time you spend in the lab and office. It depends what's going on. So if you've got a big experiment going on, you're probably in the lab for hours in a day, solid 4 hours, 5 hours, break, come back in for another solid 4 or 5 hours. And then maybe if you're writing grants to get money, you're at your desk for the same amount of time, just, 10, 12 hours a day, just smashing out as much information as you can onto paper so that someone hopefully gives you a bit of cash to do some research. So it fluctuates.
No 2 days are the same, I guess.
Yeah, there's always something new to do.
And someone else has come up with a question. They want to know whether you are currently working with universities perhaps in other countries and sharing your research with them.
Absolutely. So two of the researchers I talked about were Associate Professor Stephen Plotkin and Professor Neil Cashman, who both work at the University of British Columbia in Vancouver. And we work pretty closely with them because they're interested in the same ideas we are, but have some very useful tools that our lab doesn't have access to. And so we sort of meet in the middle and discuss ideas around both cause and cure for MND quite often. Yes, and we do work with them. We have links to Cambridge University in the UK, Edinburgh University in Scotland. We do have links to quite a few universities.
I suppose due to COVID, global conferences aren't happening at the moment. You're finding other ways to engage and to connect with researchers around the world using platforms like this. So aren't we lucky that we've got these platforms available for us to use? Let me just have a look. What other questions do we have here? Well, a question that I wanted answering, Luke, did you always want to be a researcher?
Not for my entire life.
To get towards this?
I used to think scientists were nerds. And they still are, but it's interesting. I mean, I sort of fell into it, but I can't see myself doing anything else because there's no other job where you get to do the breadth of things, where you get to have an idea and carry this idea through and see if it works or not. It's pretty unique, I feel.
Is there a standout moment in your career to date that you kind of look back on and just feel really super proud of?
Not really. They all sort of meld into one. I can't think of a standout moment. I think finishing my PhD felt pretty good. Yeah, I'd say that's a pretty good one.
Yeah, I'm sure a lot of our researchers can relate to that. Yeah. Okay, let's have a look. What other questions do we have here? So we've just got a few more minutes. Somebody here wants to know, given the similarities with Alzheimer's, has there been any success with, I'm going to butcher this pronunciation, so you'll have to correct me, cholinesterase inhibitors?
Cholinesterase.
Thank you.
All right. I wouldn't know how to answer that, and I don't know if anyone tested them in MND myself. I don't think so, but you'd have to probably think about contacting maybe the Motor Neurone Disease Research Institute of Australia. They would have a lot more knowledge about than me about what has been specifically tested in patients. We're still operating on that lower cell level at the moment, trying to build up new ideas.
Okay,
that's a good question.
And Just two more questions, I think we've got time for. One is, what's been your greatest research milestone? Is that a difficult one to answer as well?
Research milestone. I guess it's there's a lot of milestones in there. It's probably just been I wouldn't even, it's like information that's not out there or anything, but having, like, recently I had an idea and it's just been working the entire time, which is unheard of in science. Like, every time I do it, it works, and no one's had this idea before, and it's crazy that that's happening. Because often you find there's like little holes or something in your idea, but every time I try to find a hole in this idea, there's no hole. So I'm a little bit worried actually about it.
Wonderful. And just lastly, to finish off, we've got lots of comments coming in. Is there anything that the community can do to assist you with your research?
Just be aware that people have MND. Even during COVID, no other disease has stopped. Well, maybe a few infectious ones have, but really, other diseases still exist, and they're still affecting our community, impacting us and other people day to day. And there are people out here who utilizing science to try and figure out what the problem is and trying to cure it. And awareness is the first part of it. Absolutely. Yeah.
Great. Thanks. Well, Luke, thank you so much for taking the time out of your lab. I know you're incredibly busy. So really thank you so much for taking the time to put together that presentation and to answer those questions. This week is Science Week, everyone, so keep an eye out on our social media for some exciting information and updates. We've got lab teams from across IMRI taking over our social media, so there should be some really exciting things up there. Let me just share my screen so that you can link to our Facebook. So we're at facebook.com/IMRI and also Twitter @IMRI. And thank you to everyone again for supporting us. As I said already, we are a not-for-profit, so any donations that you are able to contribute would be really, really appreciated. You can make a donation at imri.org.au/make-a-donation. And also, don't forget that we have got bite-sized webinars happening all week this week. So tomorrow we'll hear from Professor Keith Aykroyd and his research into Parkinson's disease. If you haven't yet registered, you can do that at eventbrite.com.au and just search for IMRI. So once again, Luke, thank you so much for your time, for coming on board and sharing your work with us. We really appreciate it. And if anyone does have any questions, then just come through to IMRI at imri.org.au and we can answer those questions. So I will sign off. Thank you so much again, Luke, and thank you everyone for dialing in.
Thanks everyone. Thanks, Kate.
Bye.